This is always an excellent meeting. It's a relatively small meeting, a very informal atmosphere, and always plenty of time for discussion after we've reviewed the most sort of current data. So it's a great environment to start off with. There's been some really interesting topics discussed this year, and in a session of novel immune therapies, I think we really got to grips with where we are in terms of the CAR T-cell technology. And we are increasingly understanding how best to apply this technology. We're seeing greater efficacy data. We're having a deeper dive into the toxicity and perhaps the mechanisms and how we can be predictive of the toxicity. And so we had a really good debate about this and how potentially we may be able to upscale this. There are already multi-institutional studies going on, so it's going to be really fascinating to see how this technology continues to evolve. But even in the last year, the amount of new data that's been generated about this technology has been really exciting. So not only obviously are CAR T-cells of great interest, but also we're interested in the bispecific antibodies, which engage both your tumour cell and your T-cells, and technology around that. And also, again, discussion about novel monoclonal antibodies and actually some of the results that are becoming available with third-generation anti-CD20s. So a lot of data in terms of the immune therapies. The other really good thing that particularly appealed to me was that we had a great discussion about diffuse large B-cell lymphoma, and particularly the application of the cell of origin and how that may be used potentially to stratify patients for novel treatments. And really there is again an evolving story and big data sets coming in now that try and look at this and look perhaps at the difference between the general population in terms of the value of the cell of origin and the results of clinical trials to date. So that's been very interesting. We've also talked a lot about some of the new technologies and the application to lymphoma. We've talked about cell-free DNA and circulating tumour DNA, how that may potentially serve as a liquid biopsy, but also importantly being a dynamic marker for disease response and also potentially for monitoring for disease relapse, and the different technologies and really the validation of those technologies. So we had a really deep dive into those. So it's been a really fascinating meeting.